Yeah, hello and welcome everyone to today's talk about PCR-based foodborne pathogen detection. My name is Anna Jantke-Busse. I am product manager at Invitec Diagnostics in Berlin. Yeah, and for today, I'm very happy to introduce today's speaker to you, Josué Carvalho. Josué obtained his PhD in biomedicine in 2020 at the University of Beira Interior in Portugal. And today he is managing director of our Invitec Diagnostics facility in Tondela, Portugal. And since he is involved in the overall business and strategic relations in Tondela, he is a true expert in the field of food diagnostics. And today he will give us a lot of background information regarding foodborne pathogens and will introduce our newest products in this field. Yeah, and after this talk, as usual, we will have a Q&A session. So we are very happy to receive, discuss, and answer all your questions. So you can type during the talk already or after, just as you wish. So please just note that you need to type your questions to the Q&A module of the Zoom menu, since the chat function will be deactivated during this talk. Yeah, so now I would like to hand over to Josué.
Thank you, Anna. And good morning, everyone. Welcome to our talk. As Anna was saying, today we will be speaking a bit about foodborne pathogens, the problematic around foodborne pathogens, and also the products we have been developing recently to detect these pathogens. And I think you will like the solutions that we have today to present you. So, foodborne pathogens and food poisoning. This is a huge problem, as you all know. Most, or if not everyone, has faced any type of problem in the past by consuming any type of food that was not proper for consumption already. These problems may arise from physical or chemical degradation of the products, but most of the time these products arise from contamination with microorganisms, either bacteria or viruses. Worldwide, and this slide, I think it shows you how big the problem is. Worldwide, every year, we have about 600 million illnesses caused by food poisoning or contamination with any type of foodborne bacteria or viruses. Amongst these 600,000 million illnesses, these result in about 500,000 deaths yearly, and a quarter of these deaths are among children under 5. So it's a huge deal, it's a huge problem, and every year we face product recalls due to contamination or problems with people getting ill in groups. So it's a huge problem, and we have to be safe and test for this type of products. Most of these diarrheal diseases are caused by 4 particular bacteria or viruses, being that norovirus, Campylobacter, Salmonella, and E. coli are the top causes of these deaths and illnesses. Being that norovirus, as the name suggests, is a virus, and the other 3 are foodborne bacteria. So as a definition, foodborne illness is, as the name suggests, an illness that results from the consumption of food, being that most of the times the food that causes the illness has gone bad by a variety of reasons. These problems may arise from either the food being contaminated with chemical or natural toxins. When we talk about, for instance, Clostridium botulinum and the toxins that this bacteria produces, This is one type and one example of toxins that can contaminate food and then pass on through humans and cause disease. But apart from toxins, we also have the contamination with the pathogenic bacteria itself, either bacteria, virus, or parasites. So we can have a combination of all three microorganisms, and all three cause different types of diseases and symptoms when consuming food that has been contaminated. Foodborne illnesses are also known as food poisoning, because as the name suggests, the food causes any sort of poisoning to the organism and the problems that arise from there. And this contamination or this poisoning may occur before, during, and after food preparation. So that's why it's very and highly important to control all the food production chain, since the receiving of the raw material and the incoming goods during food production, when we are processing the raw material, when we are getting to the finished goods and the finished product, but also after food preparation, because there might be some introduction of a contamination or a foodborne pathogen along the distribution chain, because when we have the foods going in trucks that sometimes do not have the necessary cold chain conditions, or even when the products are on the shelves on the retailers, along the whole production chain, you can have contamination of the food product. So it's highly important that we control throughout the whole production chain. When you talk about foodborne illnesses, there are some agents that are more common than others. Here I have listed some of the most common bacteria and viruses that cause foodborne illnesses. On the bacteria side, we have Campylobacter, we have Salmonella and Listeria. These two are highly known and are two of the most common and dangerous bacteria that can result in foodborne poisoning. Then we have E. coli, and particularly the E. coli that produce verotoxins, so the stec E. coli, and particularly the serotype 0157H7, which is highly pathogenic and causes most of the foodborne illnesses that you can find worldwide and yearly. And then we also have the Clostridium botulinum that produces toxins, particularly toxins A, B, E, and F, that are highly pathogenic and can cause serious problems. For instance, in Spain, there has been a recent product recall for tortillas, where they have found that these were infected or contaminated with Clostridium botulinum, and there has been some products, some illnesses caused to tourists, actually. And there was a huge product recall in one of the most known supermarket chains, and it brought back the discussion about the importance of testing for pathogens along the production chain. Then moving on to viruses, because there are also foodborne viruses. The most common ones are noroviruses, particularly the ones belonging to genogroups G1 and G2. And we also have the problematic around hepatitis A, which is also a foodborne virus. And there are others, of course. These are just the ones we found to be more common, both common detected pathogens in food and also the most common tested ones. And what are the foods that are most likely to be contaminated by this type of bacteria or virus? Of course, raw products are always very susceptible to be contaminated because these are uncooked, unprotected, and they are almost at the disposal of the organisms to contaminate. And when we talk about raw products, we talk about mainly raw meat and poultry, and also raw eggs. For instance, if we talk about Salmonella, E. coli, Listeria, these are the products that are mostly contaminated by this type of bacteria. Then unpasteurized milk, of course, because the pasteurization is the process that we use to protect the dairy products, and unpasteurized milk is very susceptible to be contaminated. Then we also have raw shellfish. This one, particularly for viruses, is one of the top matrices that we find to be contaminated with the foodborne viruses. Then raw fruits and vegetables, also highly susceptible to be contaminated with noroviruses and Hep A virus, for instance. And finally, unpasteurized fruit juice, for the same reason as milk, as this juice is not heat treated, it is highly susceptible to be contaminated. So as you can see, you have a huge variety of products and different matrices that can be contaminated by foodborne pathogens. And of course, we have to have testing methods and test kits that are able to test all these type of matrices, providing robust results, irrespectively of the matrix and pathogen that you are testing for. Most of the times when we think about testing for foodborne pathogens, we are thinking about classical microbiology methods, mostly because all the ISO standards that are there, they are developed and meant for classical microbiology methods. So I'm talking about plating methods and doing the cultivation of the foodborne pathogens in a specific media most of the times. But when we think about more recent and more advanced techniques such as PCR-based methods, these have a lot of advantages when compared to the classical microbiology methods. For instance, if we talk about specificity, most of the media that we used in the classical microbiology methods, even though they might be specific to pre-enrich a given pathogen or a given bacteria, there's always some uncertainty if there aren't other bacteria growing in the same media. So there is a problem with specificity and sensitivity that the PCR-based methods do not suffer from, because as you know, PCR methods use highly targeted reagents, meaning the primers and the probes, and you can get a more specific method and target a single bacteria with high specificity when compared to classical micro. Speaking of sensitivity, this is unrivaled by PCR, because if you think of low limit of detections to ensure regulatory compliance, PCR offers a huge advantage when compared to microbiology methods, because most of the classical micro methods you are analyzing by an operator, a technician. Most of these methods, they involve counting and dilutions, and sometimes it's a bit susceptible to the skill of the technician itself. But when you speak about PCR, you are not quantifying, but you are measuring a fluorescence increase, and the sensitivity is not comparable to the classical methods. Then when we speak about PCR, we can also go to the differentiation of species, of strains, of variants. When we talk about pathogenic factors, that's something that you can also test quite easily by PCR. For instance, when we talk about differentiating toxins, when we talk about differentiating pathogenic traits from the bacteria, this is something that you cannot test easily by classical microbiology methods, but PCR gives you that that resource, and it does it quite easily, because you can have multiplex methods targeting different genes at the same time, and providing you with a more true answer for your testing needs. When we talk about PCR, we also talk about having a false positive and false negative proof technique. Whereas if we talk about classical micro methods, and most of the times you are working with plating, with incubation, there is a high susceptibility of having cross-reactivity or interference of the sample matrices, but also of contamination and cross-contamination. So, and PCR having all those controls implemented, built in, and having a highly specific method, these methods do not suffer as much as classical micro from the false positive and negatives problematic. Speaking about multiplexing and high throughput, definitely PCR wins over classical micro methods. You can do 96 tests in one single run with ease. Whereas if you think about classical micro methods, you are talking about doing tons and tons of plating, lots of enrichment media that you have to process. So it gets difficult when you are in a high throughput laboratory environment to process as much samples as if you would use the PCR technique instead. And then speaking of times, the turnaround time of PCR is unrivaled when compared to micro. You can have 96 results in a single run that takes between 1 hour and 30 to 2 hours. So it's a very robust and fast and safe method, if I can call it like that. So these are only 5 or 6 different advantages over the traditional methods of PCR, but of course there are more, and hopefully I will convince you today that PCR and molecular methods are the future of the foodborne pathogen detection. Usually the standard workflows for viruses or bacteria are defined by the ISO standards, and even though there might be some differences, some changes from one method to the other, usually the standard workflow is always the same. So if we think about foodborne viruses detections, for instance, usually we have a sample that might be any sort of matrix. For instance, here I've listed some of the top matrices that we test for viruses. We have the shellfish, we have fruits and salads, we have bottled water and some surfaces. That might be either environmental or even the food surface itself. The first step usually involves a concentration and recovery of the virus. This can be done by filtering the samples or by doing gradient centrifugation using some density reagents. Then once we recover and concentrate the virus, the foodborne virus, we have to have a viral control to control for the analytical process. Usually we have a virus which is related but not the same as the virus we are targeting on the food samples. We extract the RNA from the virus, and then we do a real-time PCR at the end, where we look for a target gene of interest or several target genes of interest, and we have either a positive or a negative result. If we think of bacteria, the sample prep is slightly different, because usually we have to enrich the sample in order to be able to detect the bacteria with high sensitivity and specificity. So first we have to enrich the matrix that we are testing. This involves an incubation period, usually overnight incubation period between 8 to 16 hours. Then we extract the DNA from the bacteria that have been enriched in the media. And then we use also PCR to detect the target genes from the bacteria or from the toxins, and we get either a positive or a negative result. So these are the two standard workflows. And at Invitech Diagnostics, we've been working on the DNA or RNA extraction and the PCR part of the workflows. So this is where we come in, and as I said, we have been developing in the last 10 years several products applicable to foodborne pathogen detection. And the products that I'm going to present you today are mainly nucleic acid extraction kits for both bacterial DNA or viral RNA, and then all the PCR detection kits that are compatible with these extraction kits and complete the workflow that you see here. So you start with a sample, you do the collection of the sample. Usually there's either a pre-enrichment or a viral concentration process in between. Then you extract, and then you detect the target genes. And to start off with nucleic acid extraction, I'm going to start by presenting you our RTP pathogen kit, RTP meaning ready to prep. And this kit is suitable for the isolation of viral DNA or RNA, but also bacterial DNA, even though we suggest this kit to be used for foodborne virus detection. So this kit involves a one-step lysis because it's provided with a lyophilized extraction buffer that contains all the components that you need to perform a nucleic acid extraction. So here on the right, you have an example of what the kit looks like. So you have a snapback Eppendorf tube, and on the bottom, you have a lyophilized content that contains proteinase K, a carrier nucleic acid, and also the lysis buffer. What this does is that instead of using several reagents, several bottles, and performing several pipetting steps, you end up with using one single tube where you add the sample on top of the reagents, and then you have your tube ready to process and recover highly pure nucleic acids. Currently, we have an ongoing promotion where we are offering this kit, depending on quantities, with up to 80% discount. So please contact our sales team if you are interested in acquiring or purchasing this kit, because there is this huge discount going on, and it's subjected to the availability of the kit. So if you are interested in testing or procuring this product, please contact us. As I was saying, comparing the RTP technology with the classical microcentrifuge spin-based columns for nucleic acid extraction, the benefits are huge. So here on top, you have what is the usual procedure for the standard microcentrifuge-based extraction. You see that you have to start with adding the sample, then you have to add the lysis buffer. Usually, then there is a heating and centrifugation step in between. Then you have to add your carrier RNA, then the proteinase K, then the binding buffer, and then you proceed with recovering the DNA or the RNA. If you compare with the RTP technology, you narrow down this protocol to two simple steps, which is adding the sample, adding the binding buffer into the tube, and then you have highly pure DNA after doing a couple of centrifugations in between. So this results in 40% reduced hands-on time, 60% less pipetting, and high reproducibility, because as you have less steps involved and less pipetting involved, the reproducibility that you take from the RTP technology is much higher than what you take, for instance, from the Invisorb technology, which we also provide. Then, speaking of bacteria, we recently launched the InviPrep fast lysis buffer. Perhaps some of you are still not aware of this kit, but this kit completely revolutionized the way that we extract DNA from bacteria. Because similarly to the RTP pathogen, we brought the complete procedure to a simple two-step protocol, and we can recover DNA with high purity quite fast. So this kit is applicable to isolation of DNA from all types of bacteria, and this is important because, as you know, gram-positive and gram-negative bacteria are highly different because of the cell wall, and this kit has been optimized and validated to be able to extract DNA with high performance from both types of bacteria. This is suitable for the detection of foodborne bacteria, as I said. So whereas the RTP pathogen is more applicable for viruses, the fast lysis buffer, the InviPrep, is applicable for bacteria. Usually the sample types that you can test or extract using this kit are both enrichment samples, so from pre-enrichment with media. You can also extract DNA from bacterial culture and also from colonies isolated from plates, from agar plates. So most of the bacterial samples that you can think of, you can extract using the InviPrep fast lysis buffer. And as I said, this is a super fast technology, as it doesn't require to use spin columns or any of the technology that you might be used to. And here I have a representation of the standard workflow using this kit. And as you can see on the left, starting by either a sample enrichment or a bacterial culture, or starting from the agar plate itself, with just two simple steps, which is a vorticing and a incubation at 99 degrees, and then a centrifugation, you can have 24 samples processed in less than 20 minutes. So this is, even though it's not a plate, a multi-well plate method, you can still get high throughput from this kit. And with just 3 simple steps, without several pipetting or several uses of centrifuges and different spin columns, you can get highly pure DNA with ease. So it's a very easy workflow and very streamlined workflow that you can bring on to your lab without any problems. And here is just a comparison between the InviPrep fast lysis buffer protocol with the standard and conventional spin filter extraction. So as you can see on the top, same as for the RTP pathogen, you have several pipetting steps, several centrifugation steps with some incubations in between. Whereas if you think of the InviPrep fast lysis buffer, you can do everything with just 2 pipetting steps, under 20 minutes, and a brief centrifugation and incubation at 99 degrees. So it's a very streamlined method. You can save lots of time, lots of pipetting steps, and in the end, it increases the profitability of your labs, because you are reducing the plastic consumption, you are reducing the labor time. So it's a huge advancement when we compare this method to the previous standard spin filter extraction method. Here I have some results that compare our kit with 2 other kits from the market. So we have done some testing, in this case using Salmonella as a test pathogen, and we tested our fast lysis buffer, our InviPrep, against Prepman and Kellex, which are two of the most known fast lysis resins that are there on the market. And as you can see by the PCR amplification and the PCR results for Salmonella, at either low and high concentration of Salmonella, we are able to retrieve comparable results to those of the competitor products. So meaning that this kit performs very well against the ones that are there in the market. And then speaking of different gram type of the bacteria, we have also tested several gram-positive and gram-negative bacteria. For instance, on the gram-negative side, we have Pseudomonas and E. coli and Salmonella enteritidis. On the gram-positive side, we have tested Staphylococcus, Bacillus cereus, Enterococcus, and Listeria. And as you can see from the agarose gel and the extraction band, we were able to retrieve high purity and high quality DNA from all the types of bacteria, irrespectively of being gram-positive or gram-negative. And we are able to retrieve high quantities with high purity, because you don't see any inhibition from the PCR, nor you can see any smears on the bands that can suggest that there are inhibitors or salts or any type of waste from the extraction. So that's what we wanted to show you today on the sample prep and nucleic acid extraction. And now we move downstream on the workflow, and we will present you the PCR kits that we have for foodborne pathogens. So speaking about the PCR kits, we would like to present you our InviScreen product range. So our InviScreen kits that provide you with the easy and ready to use real-time PCR method to detect either viral RNA or bacterial DNA, depending on the target pathogen that you are looking for. All our InviScreen kits are based on the Tachman technology, so they are based on probes, which provide you with a highly specific and highly sensitive target detection. All our kits that we develop in the InviScreen product range are compatible with open platform thermocyclers, as long as these contain the usual FAM, X, and Sci5 channels. So I'm talking about all the CFX96 thermocyclers, the Quanta Studio thermocyclers, also the ARIA MX. So pretty much every thermocycler that has an open platform, open software, using these three channels, which are the most common ones, you can use our kits in those instruments. And then another advantage of our InviScreen kits is that for some categories of kits, we have a single protocol, a single standardized protocol, which makes it possible to run several kits within the same PCR run. The first kit I would like to present you is the stack detection PCR kit. So this kit is used to detect the presence of verotoxinogenic producing E. coli in food, feed, and environmental samples. The most common samples that you can test for to look for stack E. coli are usually sprouts or lettuce, some vegetables, also some raw and unpasteurized dairy, some unpasteurized juice, but also some meat, and particularly raw or undercooked meat. So these are the most common matrices that are tested for stack. This kit has a high sensitivity, as it detects as low as 3 genomic units of stack E. coli. The kit is fully compliant with the ISO standard, and we also have kits available to make a serotyping of the stack. So if you know the standard workflow for stack detection and stack testing, first you test for the STX1 and STX2 genes to detect for the presence of stack E. coli. And if you get a positive result, then you have to type the bacteria and know to which O serotype it belongs. And we also have those kits for providing you with the serotyping information. The next kit I wanted to present is the Clostridium botulinum detection kit. This kit provides you with a real-time PCR method to detect for toxins A, B, E, and F, which is in accordance with the ISO standard 17919. And this kit provides you with a high sensitivity method and high specific method to detect as low as 10 genome copies per microliter. So it's a kit with a very low limit of detection and provides you with high sensitivity because of that. In terms of samples that you can test with this kit, these are food, feed, and environmental samples. On the food matrices, usually we test for green beans, spinach. We also test mushrooms. Canned food is also one of the most tested matrices for Clostridium. Also some smoked fish and smoked products such as sausages and ham, but also meat, of course, particularly raw meat. These are the type of matrices that are mostly tested for the presence of botulinum toxins. And then finally, on the PCR kits, this is the kit that we have to detect viral pathogens, and particularly norovirus genogroups G1 and G2. And this kit is applicable also for foodstuffs and environmental samples. When we think about norovirus contamination, usually we think about shellfish, we think about fruits and vegetables. Some rinsed waters are also samples that are usually tested for the presence of norovirus. And our kit provides you with a Tachman-based method to detect as low as 10 copies per microliter of both G1 and G2 genogroups of norovirus. And this method is compliant with the international ISO standard, which is the 15216. So meaning that with the kit, we also provide the viral extraction controls that are mandatory to be tested along with the G1 and G2 targets. To finish my presentation off, I would like to show you the catalog numbers to order all the products I presented today. So on the nucleic acid extraction side, as I presented, we have the InViPrep for bacterial DNA, and we have the RTP pathogen for viral DNA. Even though it also works with bacterial DNA, you can extract both with the RTP technology. And then on the detection side, on the downstream part of the workflow, we have the stack detection kits and the serotyping kit. We have the kit which is specific for the serotype 0157H7, which is the most pathogenic E. coli serotype. And then we have the norovirus and also the Clostridium botulinum toxins detection kit. All the detection kits come in a 100 tests package size. For the extraction, we have the 50, 100, and 250 prep size, so it's a more versatile ordering SKU. And with that, I finish my presentation. So thank you very much for taking the time to attend the webinar today, and looking forward for your questions and any inquiries that you might have regarding the products. Thank you. And we have the presence of Hannah's cat to finish it off.
Yeah, thank you very much, Jose, for the nice presentation. We have quite some questions for you already. So the first one is, are you planning to also develop any Salmonella and Listeria kits?
Yes, so that's a good question. And currently we are developing our own Salmonella species and Listeria mono PCR kits. So these are two of the kits we have in the development pipeline. For these ones, we will be pursuing an AOAC and microval certification because as these two methods are alternative to the classical micro methods, we have to do this for the labs to be able to accredit the method. So yes, definitely in the future you will see an Invitec Diagnostics branded Salmonella species and Listeria mono detection kit. These kits will be sold as a bundle. That's what we are planning, at least, as this will include the InviPrep kit as part of the whole solution. And also on the viral, now that we are speaking about the development pipeline, also on the viral pathogen side, we will be developing our own hepatitis A detection kit because the ISO 15216 it provides you with the horizontal method to detect both norovirus and Hep A, so it makes complete sense to have both kits available in our portfolio. So those are the three kits on the foodborne pathogen detection range that we are developing currently.
Okay, thank you. So there's another question related more to the fast lysis buffer. So, and here it's asked, do you think that the fast lysis buffer is suitable for purification of bacterial and maybe viral nucleic acids from environmental samples like natural water or water sediments?
So on the viral nucleic acids, definitely it's not suitable because the fast lysis buffer it's only validated for bacterial at the moment. We might be introducing other types of microorganisms in the future, but definitely not viral. Perhaps fungi will be included, but at the moment, what we have validated are bacteria alone.
Mm-hmm.
Speaking of environmental samples, if you are able to concentrate the sample and then filtering out the bacteria from those environmental samples, you might be able to extract the bacterial DNA from the filters. But again, this has to be validated previously to its use, but I don't see any reason why this wouldn't work for this type of samples.
Yep.
As long as you are able to concentrate the bacteria, then the kit should perform.
Yeah, I agree. So if it's about bacteria, it should work. So usually you would filter like a huge volume of water through a filter like with a vacuum, and then you would need to rinse the filter off, which you could probably do with the lysis buffer. But this, yeah, I think it should be tried, and I also believe it should work.
Yeah, I don't see any reason why.
Yeah. Here's some other interesting questions about validation, for example. So someone is asking, I am using a competitor's kit. If I want to change to yours, do I need to revalidate completely?
If your method is accredited, you might have to do a validation, a thorough validation according to the guidelines that you have from your accreditation body. The good thing about our kits is that as they are developed according to the ISO methods, the validation guidelines are usually not as strict as any other proprietary method, meaning that the specificity testing, the reproducibility testing, or the sensitivity testing for the LOD that you have to do in your lab, they can be done faster and with less experiments performed. So, of course, you will have to do a comparative study to show that this method is performing the same way as your current method, but using the ISO guidelines, you can do it faster and easier.
Okay. And also related to this, do you provide local technical support for the validation of kits?
Yeah, definitely. That's one of the things that we like to think that sets us apart from our competitors. It's that same technical support that we can provide free of charge to your laboratories. So we have a team of field application scientists that we can send over to your lab and help you on site in person to validate and implement our kits in your lab. Most of the times, the labs face some questions, some problems when adapting a kit to an already existing workflow. So we believe that it's highly important that we have this service as part of our offer. And definitely, if you have this type of needs or inquiries, contact us, please, and we will send over some scientists of our team to help you implement this in your lab.
Okay. Thank you. And yeah, maybe a last question. Do you provide also free of charge kit samples?
Yeah, definitely. If you are interested in any of the products I just presented today, please contact us, and we can arrange some samples to be sent to your labs, and then you can test it free of charge, no problem. I think that's the best way of getting to know our technology and to know that it's performing with high quality. It's to test it. So definitely, if you have these needs, feel free to contact us.
Yep. Great. All right.
There's another question, I think,
yeah.
Anna.
Yeah, is there?
Yeah, someone is asking about if the 2 extraction kits are only for manual extraction. Yes, these ones, the ones I presented, both the RTP pathogen and the EV-Prep are for manual extraction. But as you can see from the presentation, even though these are manual, they are very simple to use. They are not tedious at all. There are only a few pipetting steps involved. So even though they are manual, they are comparable to the automatic methods in the sense that you can get high throughput of it and easy of use. But if needed, we also have our product range for automated instruments such as the Kingfisher, where we provide a universal kit that also has been validated for viral and bacterial DNA and RNA extraction and works with automatic instruments. So we can also help you with that if needed.
Yeah, and just another question came up. Can we get the webinar recordings? Yes, you will get them. So soon we will send you a follow-up of today's talk, and also you will get a link there providing a recording of today's talk. All right. So then thank you everyone again for attending today's talk. We hope that you will also join our next ones. We will announce upcoming talks regularly on LinkedIn and our newsletter. Yeah, please excuse the cat again. Apparently he was also keen to join this talk.
And what's the name of the cat? Everyone is curious.
So his official name is Tarrant.
Official.
like Tarrant Hightop from Alice in Wonderland. So wow. The crazy hatter.
Yeah.
All right. So thank you everyone again, and yeah, have a wonderful rest of the day, and hope to see you soon. In case of any questions coming back later, yeah, please contact Josue directly or use our contact form provided on our webpage. So we are happy to assist at any time.
Thank you very much, everyone.